Hello class, today we're going to go over cell biology, chapter 3, Introduction to Structure and Functions of Cells. I'm Dr. Nisi Abraham. So cells are the building block of all living things, like plants, fungus, bacteria. At least you have one cells, including human body. If we want to understand how the body works, we first need to understand how cells function and how they form tissues and organs. So there are two different types of cells, prokaryotic versus eukaryotic. I named some of them earlier. Prokaryotic bacteria, example, eukaryotic human being we're studying. They have a true nucleus. Prokaryotic doesn't have a true nucleus. The simpler example is bacteria. Eukaryotic membrane-bound organelles. So we're going into that organelles. We're going to go over what are the organelles. Examples I'm going to give you. Endoplasmic reticulum, ER. Endoplasmic reticulum, one of those membrane-bound organelles. Then does have a nucleus. Animal cells are the example of that eukaryotic cells. The cell is a basic unit of life. Like I mentioned earlier, at least one cell, bacteria, unicellular. So it composed of plasma membrane and cytoplasm. That's a basic structure. Some of them have additional structures. For example, some bacteria has a cell wall, plant cell have a cell wall, but it does have that plasma membrane and cytoplasm. So which include that nucleus, cytoplasmic organelles, which is example I given endoplasmic reticulum. Another one is mitochondria, ribosomes. Those are the examples. So those cells is very small. They are microscopic. Is unaided to see with your eyes. So that's why using that microscope to see and study the structure of that cells. So we're going to see that human cell is not all the cells look same. They're not, the structure may be different, shape may be different, the number of organelles may be different, but we looking at that general cell. Then their function. Main thing you have to know from this chapter, the structure of that general cell, the organelles in human cell. We're not going to go over the plant cells. If you study plant cells, you may see some other structures like a chloroplast, big vacuole, cell wall. We don't see those things in human cells. So this based on the cells, the structure changes and their functions changes. Organelles inside the cell changes. Even in human cells, some of the cells have more mitochondria than other cells. Some of them doesn't have a nucleus. All these things we're going to see that this throughout this anatomy and physiology course. So here, this is a structure. This is a plasma membrane. Around this is a plasma membrane. That's a wall or covering of that cell, plasma membrane. That's the outer boundary. So this allow things go in and out through that, forming the interaction between another cell. Think about another cell is over here. They have to communicate. Things like that happening through that plasma membrane. Nucleus is over here. That DNA is located here. DNA is located also in the mitochondrial DNA is there, but this is mainly DNA is located here. Genetic material. Then these everything outside is called what? Cytoplasm. So it's between that nucleus and cell membrane. So inside the cytoplasm there is a structures you can see, which is the organelles. All these things we called organelles. Organelles is a general name for that, for things you see inside the cytoplasm. The cytoplasm has a jello-like consistency. How many of you guys had a jello before? So that consistency is not liquidy, it's not too solid. Then organelles, think about you had jello with the fruits inside. Each fruits representing these organelles. These organelles have different names. Thus your job is to learn this organelle's name and know what are their function. What is this organelle look differently, structurally different than this one. So their functions also going to change. So nucleus, genetic control center, mitochondria, ATP production. So we're going to see each one what are their functions. Nucleus, make sure you know that nucleus is what? Where the genetic material is located, control center, nucleolus. Inside the nucleus, see this one, nucleolus, a pink structure, makes ribosomes. Then what is ribosomes do? Ribosomes make a protein synthesis. Think if you see in my previous PowerPoint, I talk about synthesis is what? Making something. We talk about that protein formed when that amino acids put together. Each amino acids put together making that protein. That's making protein from amino acid. So that is called protein synthesis. So it is a long process. You can go watch the video, DNA to RNA, then RNA to protein. Rough ER. So rough ER, that is a rough endoplasmic reticulum. So on the wall of that endoplasmic reticulum, you see some dots, which is ribosomes. That helps to process that protein. Then smooth ER, the surface is very smooth, no ribosomes in it, makes lipid and detoxify substances. Golgi apparatus, which is this one is a Golgi apparatus, package ships protein and lipids. It is reminds me is a post office. Post office, what do? You sending that gift something to your parents. What happened? They sort them based on the size and where it has to go. That is the Golgi apparatus too. So the protein is made by that rough ER. So they packed and send it to that different parts of the cells, or sometimes it will go outside the cell. Sometimes it go from here. See these vesicles is forming inside the vesicles. Put it inside and send it to different parts of the cell, or take it outside. Then mitochondria. Mitochondria produce ATP. Then other one is the lysosomes. So this breaks down waste and cellular debris, living things, maybe foreign things, bacteria, or your own cells. Break it down by that lysosome. Think about the lysosol. Lysosol, what is that? That is cleaning or decontaminate that some bacteria or anything, those kind of things. So it's almost the name, just you can remember. Lysosomes breaking down cellular debris or waste from the bacteria, the microorganisms and stuff. Then peroxisomes, there's breaking down toxins. This is mainly dealing with the chemicals. Cytoskeleton, which maintains shape, helps movement. Give the structure of the cell. Should be somewhere here. They provide the structure. So this is endoplasmic reticulum. Is on that you see the dots, that means a rough endoplasmic reticulum. It's a smooth, doesn't have any ribosomes on it. This is mitochondria. The vacuoles, you can see small vacuoles, centrioles. That is used for that cell division time. So like I mentioned, microscope is very expensive tool to see these cells. There are different types. Mostly we use cell light microscope in our labs, compound microscope. Then plasma membrane. You remember plasma membrane is outer surface of the cell in human cell. They are actually mosaic model. Mosaic model means inside that is plasma membrane. It's arranged like this: phospholipid bilayer, 2 layers of phospholipid. This is a 1 layer, this is another layer. In between there are proteins, different types of proteins embedded inside. When you look at these things, you see different colors, maybe like that. So different colors, when you look at from outside, is a different color. It's like a mosaic model. You don't know, you ever seen mosaic floor? If you look at that, different stones on floor, so that resembles that one. So with the protein here, different proteins, some of the integral protein, some of them peripheral protein. Peripheral means only outside, only you see outside, not inside. Integral means embedded inside the plasma membrane. Then also this plasma membrane is selectively permeable. Doesn't allow everything go in and out. Selective permeable. Think about your home. Are you allow everyone to come to your house? No. It's a certain people come to your house. Whoever trusts, whoever your neighbors or something like that. So it's like that. This plasma membrane won't allow everything go in and out. That's why it's called selectively permeable. They are hydrophobic and hydrophilic region. Hydrophilic means hydro means water. Philic means love towards the water. So water attracting phospholipid head part. So it's look like this. Head part is what? Hydrophilic. The tail portion is hydrophobic. Phobia means fear. Doesn't like water. So if it doesn't have water, is going to be inside of that plasma membrane. So this everything outside is a extracellular membrane. So it does have water. Fluid is here, fluid is over here. So this hydrophobic region is actually embedded inside the plasma membrane. Then you have to know that membrane transport. Like we said that things everything won't allow to go in and out. So some of things cannot go through this. Some of them is too big. Some of them may be hydrophobic, or certain things reason it cannot go in and out. So what happened? There are different ways things go in and out. Types of movement we're going to study. One is a passive. Passive can be diffusion or osmosis. Diffusion is the one the molecules will go in and out. Then that's a passive. Because of passive, it doesn't requires any energy. It can go in and out without energy requirement. So if you osmosis, that is the water will go in and out. Water will go in and out. So diffusion is chemicals go in and out. Water will go in and out through that the process called osmosis. Think about spraying a perfume in a certain place. If you give some time, what happens? The perfume will diffuse into that wherever that from the higher concentration to lower concentration. So the diffusion is the one going from that higher concentration to lower concentration. Is going water is moving from higher to lower is osmosis. Chemicals moving from higher to lower is the diffusion. Then facilitate that movement. This is by protein channels. Some protein channels here allow the things will go in and out. Those things cannot go through the plasma membrane, so they allow things to go through that channel. So for example, you have a wall around your house, then you have a gate. Gate will allow things to what? Bigger stuff to go in and out. Active means what? It requires ATP. ATP is what? Energy. So when you do active transport, it requires energy. Then vesicular transport, there are two ways. Vesicular transport means the is forming vesicles, like I mentioned earlier, Golgi apparatus, they forming vesicles. They merge into that cell plasma membrane. So it will, whatever it is, exit, release into the cell, or it will exit out. This is called endocytosis or exocytosis. Endocytosis means the things will come into the cells, or exocytosis, the cells going out from the cell. So these are different types of transport mechanism happening in our system. This is very important for when you take in that nursing exam and things, tonicity, what it is. Osmosis and red blood cells. That's one of the scenario questions you can get from this topic. Isotonic, hypertonic, hypertonic. So isotonic, no net water movement. Hypertonic, water enters cell swells. We're going to explain what it is. Hypertonic, hyper means what? More, higher. Hypo means what? Below. So in this case, if you think about it, take a beaker. So we're going to put that red blood cells, RBC, all of them. This RBC. So think about this concept. This is inside the water, okay? It's water. This is the liquid, whatever the solution, it does have a 50% of concentration. So also inside the cell, it's also 50% of concentration. That means they both are what? Same concentration. So it's a net water movement has to be equal. Will go in and out easily. So it's nothing going to happen, which is called isotonic solution. Here, this one, another scenario, I'm going to talk to hypotonic. Hypotonic solution. Hypotonic means this one has 30% of concentration. This have so 30 means what? So 70%. Which one is more concentrated? This one. So this solution is what? Less concentrated. So the more water inside here. Is this a solute concentration we're talking about? So the water will move from here to here. So what happened? These cells are going to be swells because a lot of water is rushing inside because it's a more concentrated of that solute. So water rush into that one. Finally the what happened? This is going to burst. Okay, this is burst. Then this one talking about hypertonic. Outside the cell has more concentration. That cell itself is less concentrated. This is a hypertonic solution. So what happened? There is a liquid from the cell going into that solution around. So this cell become what? Everything water is losing, cells be shrinking. Okay, both is not a good. Sometimes your swelling happens. Water accumulates certain cells and around, so it can burst. The cells can burst. So cytoplasm, I mentioned that inside the plasma membrane, outside the nucleus, cytoplasm has what? Cytosol and organelles. Two things in the cytoplasm. Cytosol is a fluid or with the enzyme, ion and nutrients except that organelles. If you think this is a mitochondria, this is organelles. This is all this liquid, fluid, semi-fluid structure which is a cytosol. Then if you look at that nucleus, they have a nuclear envelope. Nucleus, nuclear envelope, nucleus inside. They are double membrane. They have nuclear pores. I want to see if I can erase. No. So it does have nuclear pores. So allow things to go in and out. This nucleolus makes a ribosomal RNA. Ribosomal RNA. Then it does have chromatin. Chromatin is DNA with the protein. So it's like a DNA is a long strand. If you see that other PowerPoint I mentioned, long structure is coiled around the protein. So proteins like that, this things coiled around it. It's coiled around. So it can condense inside the nucleus. So this genetic material, DNA is inside. DNA is a double stranded. It won't go through that through nuclear pods. What happened? The DNA is converted into RNA inside the nucleus. Then RNA is single stranded, will go outside the nucleus. So this is a process transcription. We will explain transcription later slides or PowerPoints. So we have a few things students confused to chromatin, chromatid and chromosome. They are DNA. Everything is DNA is in the nucleus, different forms, different timing. You see these things. Chromatin, the loose DNA in the nucleus, not coiled around the protein. It's going to be, if you see that nucleus, this is you don't even see that. When you look at that cells, you don't see DNA clearly. So that is what loose DNA DNA in the nucleus, chromatid, one half of duplicated chromosomes. That is called chromatid. So we're going to see that once replication happens, DNA replication, for example 46 chromosomes we have it, 23 pairs, 46 chromosome human, this become that 92 one that DNA replication happens. So that is that's a double of this one. That's very important. That is called the part we call the chromatid. So the chromosomes condense DNA during division. So this DNA coiled around the protein So at this point you will see that condensed DNA during that nuclear division. Then cytoplasmic organelles, I mentioned that, you can read it. Ribosome, what they do, protein synthesis, ER, two different types, smooth and rough, making protein. Smooth is making lipids. Golgi modifies or the protein. Lysosome digestive enzymes there, so you can digest, break it down, cell debris and things. Peroxisome detox, dealing with the chemicals. So mitochondria is ATP production, which is energy, provide energy. Proteosomes, protein degradation. Some proteins malfunction, it doesn't form the way it's supposed to be, so it's a degradation happen through that proteosomes. So here's surface structure. So then other things, centrioles and spindle fibers, this forming the cell division type. So here in that cell nucleus, they should have something called centrioles. Pair like that, you see that. So it will go into that divide and go into that different poles when that cell started dividing. It's going to different poles. So that is the time of that cell division. It's forming that this is a fiber formation happen here. So this fiber is forming. The chromatid is actually attached, sister chromatid is attached to that fibers. So for that nuclear division type. Microvilli increase the surface area. So these cells sometimes they have microvilli, villi, and microvilli is a projections that is increase the surface area of that cell and structure. Because think about it's only this way, its the surface area is this much. Now it's like this projections, you can increase the surface area. Mainly you see that where the absorption things happen, digestive structures, those cells have these kind of microvilli. Then we're going to go over the cell cycle. So the word break it down, cell and cycle. Cycle is what? Starting one point and around that. So there are different phases of cell cycle. One is G1, S phase, and G2 phase. G1 and G2, which is a growth phase. S is a synthesis, DNA replication. Happens. Okay. So mainly G1 and G2 phase cells are growing, growth phase, and make sure do their functions. that's whatever the cell is for, that's a function. Here DNA synthesis, making the DNA replicate and making that a copy of that DNA. So that's why we have 46 become 92 after that DNA replication. Cell division. In the cell cycle, most of the time cells spend time %90 time interphase, which is the one we seen that here this is a little bit wrong in this. So this G1 we talk about the growth phase and do their function, ready for the S phase where is that DNA replication happened. From after DNA replication is a G2 phase, is a growth and ready for that cell division. So the cell division, we're going to see that this is a mitosis I'm going to talk about. There are different types of cell division. Mitosis is a somatic cell division. Somatic cell division means every cells except your sex-related cells, which is ova for the female and male is a sperm. So those two actually go through the different types of cell division. Somatic cells, epithelial cells, connective tissue, connective cells, all these cells, somatic cells go through that is mitosis. Mitosis, one of the thing about mitosis, one cell divided into two new daughter cells. This one is a 2N diploid 46 chromosome. This is going to get a 2N 46. This I'm talking about human, okay? 46 chromosome. That's the way. If you're looking at that other cell division, that if meiosis, parent cell 2N, make it into haploid cell. This diploid is a haploid cell. This is a 46. This is only have a 23. Because it's if it's a ova 23, your mother give you 23 chromosome from her. Your dad provide 23 chromosome, another one. So the sperm and ova fertilized and fused and forming that 46 again. So that's why it's only 23 for that sexual cells like a sperm or that ovum. Then this nuclear mitosis, karyokinesis, this is a nuclear division, that nucleus start dividing for the cell. Inside the nucleus, for example, I will write